So we're going back through all of chapter 8. So I'm not starting with all of the researchers, but let's go. So a nucleotide is one sugar, and I need to write on here. So that's one sugar right there. Let me change the color. Let's get some white going on there. So one sugar, which you already know, I'm sure you remember, is deoxyribose. One phosphate. And one nitrogen base. So this is a nitrogen base. This is the basic unit of DNA. So I know that comes up on your test. It will say the basic unit of DNA, and you will say nucleotides. Going to the next slide here. All right. Now there's 4 bases. T stands for thymine, and it matches with adenine. And like I said, straight letters go together, curvy letters go together. So C stands for cytosine. And G stands for guanine. Yep, you're going to have to know those. So here we go. Okay, so now what we have is DNA is a double helix, meaning there's 2 sides. So we've got one here and one here, and it spins around each other. And if you take a look, we've got adenine and thymine matching together. So we got that right there. And then we have cytosine and guanine. So that's the base pairing rule. Straight letters go together, and C and G, curvy letters, go together. Okay, what's this CA plus CG? So how would we figure this out? This is just basic math, but the number of A's is always going to equal the number of T's, and the number of C's will equal the number of G's. So if you add these 2 together, you're going to get those 2, because they should be equal, unless we have mutations. Okay. So here we go. Replication, and I know you can't see this because my little thing is here. This should say DNA polymerase. Okay. Helicase, which is an enzyme, unwinds the molecule. Because remember, it's a double helix. It's all twisted. So what we need to do is, so that it doesn't get all tangled, we have DNA, we have helicase that's going to go in and unwind the whole molecule. Now that it's nice and straight, we have polymerase. So see, it's all nice and cleaned up and straight. Polymerase goes in and adds the bases so that we will get 1 strand and 2 strands, and they will be absolutely identical to the original strand. Is that important? Yes. This happens during the S phase of the cell cycle. This is really important. This is how you make more cells. Okay. Now, polymerase also proofreads. So it makes sure that we're putting the right bases with the right bases. No C's with T's. No, we want A's with T's. All right, so now, so replication means making an exact copy of the DNA, versus transcription, we're going to make some mRNA. So look at right here. The DNA is split open, but only one side is used for our template. Well, how does your DNA know to do that? Because there is a code in it, which happens to be AUG, which is, like, it's not setting up right here, but it is AUG, which is methionine. Okay. Remember, when we're making mRNA, there is no thymine. It's uracil. And it's going to be only 1 strand right here. Whereas DNA is 2 strands, RNA is only 1 strand, and it has uracil. So if ever you see that U, we know it's a strand of RNA. So as we go here, it's called messenger RNA because it has the message. It's the recipe to make protein. Let's go, protein. Woo! So our nucleus is right here, and I know it's cut off. You can't see it. But the messenger RNA leaves to go to the ribosome, is what would be going here, to make protein. So in here we have, if this comes up on your test, which you know it will, in the nucleus we have replication, which means making an exact copy of the DNA, and transcription. Transcription is making the mRNA. But that can't stay in the nucleus. It has to leave to go to the ribosome. Because why? What happens in the ribosome? That's where we make protein. All right. So now I had you do this in class, and some of you were here, some of you weren't. I had people walk over to the board carrying little signs, and it had 3 letters on it. So if we go back here, okay, oopsie. If we go back here, 3 letters on the DNA is called a triplet. 3 letters on RNA is called a codon, and I'm going to make that a different color, and I'm going to make it thicker. Okay, so 3 letters there is called a codon. Well, what we want to make protein. This isn't protein yet. So we're going to leave here. And here's the codons. This is a codon. Remember, this is the mRNA. It says it right there. This is the anticodon. This is what's going to read the codon. So this is the anticodon. And look what the anticodon, the tRNA, is hooked to. It's hooked to an amino acid. So look at this guy's missing his amino acid. Oh my gosh, what's he going to do? He goes and matches with another amino acid so that we can make the correct protein. Does it ever happen where the tRNA matches with the wrong amino acid? Hopefully not, but of course you know it does. That would be a mutation. All right, so this is the table so that we know what amino acid goes with which tRNA. This is the table that lets us read it, okay? And you start at the middle and you work your way out. So let's do this together. All right. So here we have our single strand of DNA. Awesome. Okay. So it says transcribe. Remember, there's no T's in mRNA. There's U's. So this would be CCU. This would be GAU. AAC. CGG. AUU. GUC. Beautiful. I just transcribed it. Now remember, we'd go to the ribosome now. Ribosome. We got to go to the ribosome because that's where the magic happens. Now we need to translate into tRNA. So I need to write the tRNA structure because that's what's in here. Okay? So this is going to be GGA. Wait a minute. Doesn't it look like the top one? Mm-hmm. And this is going to be CUA, because remember there's no T's. And then we have UC, and then we have CGG. Oops, I don't know why I wrote that. That's just not even right. I'm going to erase those. All right, so boop, boop, boop. It's going to be GCC. I copied the wrong one. And then this one is going to be AU. Why? I copied it again. Darn it. This one's going to be UA. And then this last one is going to be CAC. Now we need to write the proper amino acids. I'm going to change my color. I'm going to do red. Why? Because I feel like it. That's just what we're going to do. So GGA. So we start here. We go GGA. And that's going to be glycine, GLY. And then we're going to go CUA. CUA. Oh, wait. I don't know why I did that. So, yep, I goofed up. CUA. That's leucine. And then UUC. UUC is alanine, phenylalanine. All right, so that's phenylalanine. So we're going to put just FY right there. And then we have GCC. So GCC is just alanine. We're going to do ALA. And then we have UAA. UAA. Oh my gosh, it's the magic right there. And look what it says. It says stop. So well, wait, what about this guy down here? It says stop, and that's where this protein would stop. The amino acids would hook up, and we would have a protein. That is all of it in a nutshell.